INDs with the FDA for clinical trial. So we're very collaborative. We created an open data portal that I'll show of the early. My name is Matthew Hall, and I'm a group leader of biology at NCATS and also the acting director of the early translation branch. And it's an honor to present a talk to you today about our response at NCATS, part of the National Institutes of Health, to the COVID-19 crisis, and how we've used drug repurposing libraries and created an open data portal that I'll share with you today. And I think this talk will align very closely with the meeting that you have on open platforms for translational research to celebrate the 20th anniversary of the Korean Chemical Bank. I was honored to host visitors from the Korea Chemical Bank last year, and I was very excited to visit you in Korea. I'm sad that I'm here in America and I'm not enjoying the food and the company and the science, but I hope I can visit very soon and we will begin collaborating together. So at NCATS, we are a translational science research institute, and we are really an integrated pipeline for small molecule discovery. My branch is involved in early discovery, and as I will tell you, we collaborate on creating and developing small molecule probes for new targets in biology to share with the scientific community. To do this, we're structured scientifically more like a biotechnology research institute. We have research groups in assay development, high throughput screening, medicinal chemistry groups, and these work together to develop teams, develop molecules as a team, as tools for biology, and sometimes they go into preclinical development for diseases of interest as well. And at NCATS, we can take a small molecule, if it's important and relevant, all the way through to file INDs with the FDA for clinical trial. So we're very collaborative, and our goal is always to collaborate with deep experts. So in an ideal project, a domain expert in a disease will say, you know, I have studied this disease and I think there is a gene that is important. I have a therapeutic hypothesis, but I don't have the probe to test my therapeutic hypothesis in cell models or in animal models. So our goal is to collaborate and develop probe molecules to test these therapeutic hypotheses. We're always collaborating, and we always focus on developing new probes for understudied targets, first-in-class probes, and new tools for the biomedical research community. We try to focus on targets that the pharmaceutical industry is not studying. And we worry and think a lot about target validation as well. Are we starting the right projects and the right collaborations where we have confidence in the research of our collaborator? So we're not a fully fledged drug development program, but we do look for opportunities to partner our science, and we hope that by creating probes and testing therapeutic hypotheses, this is a launch pad for drug discovery programs in the private sector. So, as I mentioned, we through our collaborations, we develop assays, and we use high-throughput screening with large small molecule libraries to discover starting points for medicinal chemistry. We have a large automated platform for high-throughput screening with multiple automation arms that can interact together. They are surrounded by all of the instrumentation and equipment needed for a bioassay. We screen in 1536-well plates, so very low volume plates. And we always screen in dose response. So most places will screen with a single concentration of compound. 10 micromolar is very, very popular screening concentration. But we like to screen in dose response because we can rank best and weakest compounds in screening. So this is important, or more important. But also, as well as ranking the efficacy of compounds, it makes sure we don't miss. We screen up to 50 micromolar. So a very weak hit you might ignore at 10 micromolar, but it could be interesting, and we have examples where very weak compounds can be developed. At 10 micromolar, two good compounds will look the same. When you do dose response, you see a big difference in activity. So we can screen over 500,000 compounds a day, and we have a number of library approaches. We do, as I will tell you later, a lot of drug repurposing screening, and we use annotated small molecules, libraries with known mechanisms of action for each molecule, a pharmacologic equivalent of a functional genomics screen. We have about 10,000 molecules that cover FDA-approved drugs, experimental drugs that were never approved, probes with known mechanisms of action, natural products. And we regularly use these to look for repurposing opportunities, especially in the rare disease space. And there are over 7,000 rare diseases. Most of them do not have a therapeutic or a drug, and repurposing is a great opportunity. But as well as our annotated small molecule libraries, we have over 200,000 molecules in our diversity library of small molecules that are diverse chemotypes that are useful or amenable for medicinal chemistry diversification. And this is really where we do a lot of our discovery screening. We're expanding our library regularly, and actually we now have about 250,000 molecules. All of the screening takes place at NCATS. Traditionally, we do not send our libraries out to other places. So people collaborate with us, they bring their assays, and we conduct our screening in our industrial screening platform. So we do a lot of science, and we do it at scale. And we've generated a lot of data. Recently, we had a review, and we had to summarize 4 years of accomplishments. And so you can see that as researchers and collaborators, we've been running almost 1,000 assays every year at NCATS. Some of them one plate, some of them many plates, full high-throughput screens. In terms of the types of biology we interrogate, a little bit over half of our assays are cell-based. And therefore the rest are mainly biochemical assays, enzyme assays, and a small number of organism-based assays, things like malaria or a tissue plate. And in the last 4 years, we've generated over 131 million wells of data in our assays, 85,000 plates. So that's a lot. And all of the data we generate, we share openly through platforms such as PubChem, which you'll be hearing about, I think, in one of the other presentations at your meeting. Along with our assays, we do a lot of chemistry. We have about 80 active medicinal chemistry projects in any year. Some of them are very active, some of them have a little bit of activity. And because of all of these projects, our medicinal chemists make about 1,700 new compounds a year. A lot of those new compounds eventually are actually folded back into our screening libraries. So we do screen compounds that we produce in our medicinal chemistry programs, and sometimes we find interesting second activity for compounds we made for a different program. To make this many compounds, our chemists perform over 5,500 chemical reactions every year. And so this is a lot of chemistry. It's large scale, and the goal, as I said, is using this medicinal chemistry, testing against assays to create optimized probes. We do a lot of work at NCATS to share information about drug discovery, drug development, assay development, high-throughput screening with the wider scientific community. And we have a program called the Assay Guidance Manual, and you can see a link here. It's a free book, electronic book of over 1,000 pages. We have an editorial board of public and private members from pharmaceutical companies, government, academia, and they share their knowledge and help make sure that the Assay Guidance Manual is up to date, and it has chapters on different types of assays and different assay approaches, and important aspects like reproducibility, data analysis, and ensuring high-quality discovery science. Because as we know, many drug development programs fail because they can't reproduce the original science. And so the Assay Guidance Manual is a resource that I would encourage you all to look at, where we try to share our knowledge of probe and probe discovery. So I am not there because of COVID, so I will tell you about COVID. When COVID started, and it was very clear it would affect us in America about February, we began to plan, how can we respond to this? We're a translational science institute, and it's an interesting challenge. All the way through the drug development program, we have challenges because this is a brand new disease. In 2018, 2019, this disease didn't exist. So at the same time, we need to build a pipeline for discovery. We need to understand the targets. We need to build assays with those targets. We need to have the right libraries to do screening against those targets, and we need to have good preclinical models. And we have to do all of this at the same time. So really complicated translational science. And of course, drugs take years to make. And so drug repurposing became a very popular approach. So we knew that ultimately, and I believe still, that vaccination will help us to eradicate and manage COVID-19. But we knew that when this disease was 2 months old, we had an immediate need for treating patients and that drug repurposing was going to be a path. But we also wanted to make sure that we openly shared as much science as possible. And so we knew a lot about COVID-19 from other science. We knew, and from SARS and MERS and other beta coronaviruses, and confirmed that it binds to the ACE2 receptor. It enters the cell like all viruses. It's unpacked. The RNA, it's an RNA-based virus, is replicated. There's protein products translated as well, and it's repackaged and eventually leaves the cell. So there are many opportunities to drug the virus, and there are many opportunities to drug the host cell. And so the way we thought we would do this is by taking drug repurposing as our approach. So in 2009, we created the NCATS pharmaceutical collection. It has almost 3,000 drugs approved for use in the United States, Europe, Japan, Australia, and Canada. And we must check Korea, that we may be missing some unique Korean drugs. And we update this every year. And you can learn more about any of these drugs at this site, drugs.ncats.io, at our site called Insight. And actually, since we created this library, we've screened it over 800 times. So we have a lot of information and data to share against SARS-CoV-2. The attraction of drug repurposing is that a normal drug development pipeline, you screen hundreds of thousands of compounds. It can take 15 years. It can take billions of dollars to create a drug. We may have billions of dollars, but we don't have 15 years. And so drug repurposing is great because we made the library. You can screen new assays, and if something works, you can very quickly go into a clinical trial because you understand the toxicity and you understand the dosage from the original indication that that drug is used for. If you'd like to learn more about this library, we just published a new paper, a 10-year review of the pharmaceutical collection. So we knew many labs, including in Korea, are running experiments. They're doing screening, but we don't know the assays and all of the details. Most of the data is not shared, and we felt that this would impact and delay how quickly we can bring treatments to patients. Maybe if we can bring all the data together and make all the data available, people would find opportunities for drugs that may be not number 1, they may be number 10, they may be number 50, but it might be number 10 in every single assay. It might be a great opportunity that we miss by doing one screen. And so we decided that we would, at NCATS, take as many as targets and develop as many assays as we could, screen them against all approved drugs, and make our data available to the world. And in fact, make it available to the world before we publish. So we do screens, we generate the data, we confirm that it's reliable, the assay worked well, and we put the data on our webpage that I will show you so that it can be downloaded and accessible all over the world immediately. We publish later. It's more important to share the data. So what were we going to assay? So we look at the viral genome, and there are all of these genes, many of which we can guess what they do from our knowledge of other viruses. Some, we don't know so much about their function. We still don't know so much about their function. But we looked at the genes from a viral point of view that we had a lot of knowledge about, and we started developing assays. Some of these we know quite well. RNA-dependent RNA polymerase is the target for remdesivir. But we have a helicase, we have the spike protein, which interacts with ACE2, very important, nucleocapsid protein. So we've been developing detection assays, activity assays. We also have many cell-based assays, including pseudoparticle for entry into cells, live virus assays. We use other BSL-3 laboratories, and we send them our libraries, and they generate the data and send the data back to us. We do not have a BSL-3 laboratory at NCATS. And human targets. So I mentioned ACE2, but also important targets like TMPRSS2 that we are learning are important in viral entry or viral maturation. We tried to develop assays against these, and we've been screening them. And so we want to make sure we share this data globally as quickly as possible. And so to do that, we created the OpenData portal. This page was made especially for COVID-19. We generated the website very quickly in just a few weeks, and we started uploading data. And as I'll show you, we have a data browser to share all of the data that we generate, information about all of the assays that we screen, animal models. We don't have animal models for COVID-19, but we've been working at NIH on capturing and cataloging all animal models, and you can access that information. Summaries of genomics efforts. We have highlights and blog posts and resources that are available to people who want to learn more about high-throughput screening, data analysis. And so as data is generated, we uploaded it to the website quickly, and you can access this link to look at the webpage. When you click on the OpenData portal, you see a list of all of the assays, and as we add new assays, we increase the page. And we color code activity. So green is very active. We have counter assays, we add that data. So purple is activity in a counter assay. And you can see on the left here that for all of the thousands of compounds we've screened, including our approved drug collection, you can list all of these here and take a look at each of them. If you're interested in one drug, you can type the name, click on the drug, and learn more information about that compound. Just as important as the data is the assay. And so when you click on the assay tab, we make all of the protocols for all of our assays available before we publish. So any lab in the world can look at these assays, reproduce them in their lab, and use them for drug development programs in the private sector, in the public sector. We want to make them open and available as quickly as possible. In fact, when you click on an assay, you can download a protocol PDF with step-by-step instructions for every single assay, description of the assay. If it's published later, we add a link so people can learn more from the manuscript. But the goal is to share this information with the scientific community as quickly as possible. But more importantly, you can look at the assay and the details, you can download the protocol, and you can download all the data. So you can look at the data, but for each assay, you can click a button and download all of the drug repurposing screening, all of the dose response data, so that data scientists, informaticians, and biologists can work with this data on their own platforms in the way that they prefer to interact with our high-throughput screening data. Again, to try and be as helpful and useful as possible for the global COVID-19 response. So, as I mentioned, our open data portal is arrayed so that people can browse through the data, search for a compound, browse through different libraries, and all of the data is widely available to the community. And we've had thousands of people visit and look at this site. You can go and look at this site. We've had a lot of attention. We did release a preprint that describes the portal, and we're hoping to publish that soon. It explains how we design the portal, the goals for data sharing, and gives something for people to be able to cite, because we're putting unpublished data on the webpage, and we realized that people said, how do we cite your webpage? And so this explains the philosophy of sharing data openly, sharing the biology openly to make sure people can reproduce our data. People have been downloading the data. This is just one example. This website uses all of the NCATS data to use artificial intelligence predictions for new drug ideas. Dr. Francis Collins, our director, and Dr. Tony Fauci, the director of NIAID, who is a face of COVID-19 at NIH, have been speaking about our portal. And so people are aware that if you think, I wonder if that drug might work against COVID-19, you can come to our portal and you can check. You can see that chloroquine is not so good, for example. Other drugs work very well. Remdesivir is very good. People can contact us. They can submit compounds for testing as well. We've been testing other people's compounds. And as I said, animal model data and information is available. So if you want to know what the latest and best animal models are that the scientific community is embracing for testing in vivo efficacy, all of that information is there. So we made the portal to share NCATS data, but we need your data too. And the second part of this, as we generate our own data, is that we're speaking to translational science and high-throughput screening groups all over the world, and we will be hosting their data. Groups from China, Europe, Canada, United Kingdom. we're speaking to and reaching out to all of these screening centers to say, if you're doing drug repurposing screening, please contact us. And when you've published your data and you're comfortable, we will make a page for your assay protocol. It will link to your institute, it will link to your paper, and we'll share all of your high throughput screening data as well. And so by putting all of our global data side by side on this OpenData portal, people will be able to seek and find new opportunities and new ideas for treating COVID-19. And hopefully we don't have this problem, but this data will be available and these assays will be available for the next COVID-27, the next global pandemic that involves a beta coronavirus. So this is really a long-term initiative. So we can ask, do the same assays give the same results at different places? Are there molecules that are active across many assays and we wouldn't find them? And so please do contact me and reach out to me if in Korea you are screening and doing drug repurposing screening on COVID-19. We would love to work with you to share your data with the global community. I want to thank you again for listening to my talk. It is 21 minutes, not 20 minutes. I'm very sorry. I thank all of my colleagues at the National Center for Advancing Translational Sciences, and I really thank you for listening to my talk. And I hope we can meet face-to-face soon when COVID-19 is over. I look forward to collaborating. Thank you.
Good morning.
Thank you for your talk. Hi, Matthew.
Which is
Hi.
U.S. National Library of Medicine. NCBI is the home of many resources, including PubMed, PubMed Central, clinicaltrials.gov, GenBank, and good morning.
Hi, Matthew. I wonder what time it is there.
It is 9:30 p.m.
Thank you for the talk in late time. And some must have some questions, so we will give you some questions. So 우선 여기 참석하신 분들께 그 질문을 받고요. 그리고 그 다음에는 온라인으로 받은 질문을 드리겠습니다. 네, 혹시 질문 있으신 분? 네, 질문이 없으시면 we have a translator, so she's gonna give you a question.
So hello, thank you for joining us at today's symposium. So we have collected questions about your talk in advance, and I'm going to translate those questions to you. Please speak at a measured pace, speak slowly.
I will.
And if possible, please pause after a couple of sentences. So first question is there any reason that you screen all compounds with multiple concentrations?
Yes. We test multiple doses, and that helps us find the most potent compounds. When we screen at one concentration, two compounds are active, they look the same. But when you generate a dose response curve, you can tell which compound is very active, which compound is weak. And that helps us to prioritize for further testing, such as animal studies.
첫 번째 질문은 이 스크리닝을 진행을 할 때에 여러 가지 용량의 화합물에 대해서 하나의 용량이 아닌 여러 가지 용량의 화합물에 대해서 스크리닝을 하는 이유가 무엇인가에 대해서 질문이 있었고요. 예, 그렇게 하는 이유는 이렇게 다양한 용량에 대해서 우리가 검사를 해봄으로 인해서 화합물 중에 가장 강력한 화합물이 무엇인지를 찾기 위해서 그렇게 하고 있습니다. 이런 방식을 통해서 용량 반응 곡성이 나오게 되는데 이 중에서 어떤 게 활성도가 가장 높은지, 그래서 가장 강력한 화합물을 찾아내고 그것을 먼저 선택을 해서 이후로 예를 들어서 애니멀 테스트 같은 것들을 진행할 수가 있습니다. So thank you for your answer. And the second question is, do you get any feedback on your research from pharmaceutical companies or industries for clinical trials?
Yes, we do. We collaborate with pharmaceutical companies to develop new assay technologies for discovery. And sometimes the probe molecules we develop are licensed by biotech or pharmaceutical companies for further development. and NCATS can take compounds to clinical trials. We share information and understanding so that we understand their challenges, the problems in drug development they are trying to solve, so that we can try and develop and invent new techniques to solve their problems. So it's an active conversation.
두 번째 질문은 이제 임상 실험과 관련해서 제약사로부터 지금 진행하고 있는 NCATS에서 진행하고 있는 연구에 대해서 제약사로부터 피드백을 받는지에 관한 질문이었습니다. 저 답변을 하시기를 이제 제약사들과 긴밀하게 협력을 있다라고 말씀하셨고요. 특히 SA 기술 같은 것을 개발을 할 때 협력을 하고 있다. 그리고 이제 그 탐침을 위한 프로브 몰리큘을 만들 때에도 제약사에서 라이센싱을 준 그런 것들을 사용을 하기도 하고 또 이 NCATS라는 기관에서 임상 실험을 위해서 화합물을 제공을 하기도 합니다. 그래서 제약사들이나 아니면 바이오 기업들과 적극적으로 정보를 공유를 하고 있고요. 이러한 방식을 통해서 제약사들이 신약 개발을 할 때에 어떤 애로사항이 있는지를 우리도 이해를 할 수 있고 그래서 이러한 제약사들이 신약 개발을 하는 데 저희가 많은 도움을 줄 수 있습니다. And this is the last question. Okay. You started combination screening. Is there any strategy to design combination of compounds?
Yes. Combination screening requires more resources, more plates. We test 6 concentrations and 6 concentrations for 2 compounds. So one combination takes 36 wells. So you can imagine it requires many plates. Our standard approach is to test one compound against all approved drugs to identify synergy, but also sometimes to identify antagonism, blocking, so that you cannot give 2 drugs together. In COVID, we have been doing this, looking for synergy between antiviral drugs. Historically, people just use this approach for cancer, for killing synergy. But we have applied it to antiviral research as well to try and identify good synergies for clinical trials for COVID.
So I was just informed that I don't have to translate your answers because your talk wasn't translated either. So once again, thank you very much for your participation today.
Thank you very much. It was an honor.
There is one question from the audience.
Okay.
궁금한 게 지금 어쨌든 미국 정부 세금으로 이제 기관이 운영이 되는데 어쨌든 거기서 확보된 데이터는 이제 전 세계에 다 이제 공유를 해주시는 거잖아요. 굉장히 제너러스하게. 그러면 이제 미국 국내에서 이제 뭐 미국 국내 회사나 미국 국내 기관이 도움을 받을 수도 있지만 이제 외국에 있는 기관이 그로부터 많은 혜택을 받을 수도 있는데 그런 부분이 미국 정부에는 그게 잘 설명이 되는 것인지 제 질문 이해가 됐습니까?
네. So this program, your research is funded by taxpayers' money of the U.S. citizens. And the research outcomes are shared by the international community. Of course, it may benefit U.S. local companies, but it can also benefit companies of other countries or organizations of other countries. How does that help pursue your government to continue to invest and provide budget support using taxpayers' money?
So our mission is to improve human health. And we conduct research that improves human health all over the globe. And that is a good thing. So we don't have any problems justifying the importance of our work to the taxpayer. And when national crisis occurs like COVID, that's another opportunity and a reminder that the work we do certainly benefits American taxpayers, but it benefits all humans.
Thank you again.
Thank you.
Thank you for your talk, and I really hope to see in real world. And let's move on the next speaker. Bye.